Ladies and gentlemen, welcome to today's webinar, Myeloma Vaccines, brought to you by the Multiple Myeloma Research Foundation and sponsored by AbbVie, Amgen, Bristol Myers Squibb, Celgene, Genentech, Janssen, Karyopharm, Novartis, and Takeda Oncology. It's my pleasure at this time to turn it over to Mary Jerome at the Multiple Myeloma Research Foundation, who will begin the webinar. Welcome, Mary.
Thank you, Sandra, and welcome everyone. I'm Mary Jerome, Director of Medical Communications and Education at the Multiple Myeloma Research Foundation. And I'd like to thank you all today for joining us for this really exciting webinar. I'd also like to thank our sponsors, AbbVie, Amgen, Bristol Myers Squibb, Celgene, Genentech, Janssen, Karyopharm, Novartis, and Takeda Oncology for providing us with the educational grants to support this webinar. First, today, a little bit about us. We're a patient-founded organization. We started in 1998 when Kathy Giusti, our leader, was diagnosed with multiple myeloma. We have a keen sense of urgency and a laser focus on extending patients' lives, since we know that there's no time to waste in our quest for a cure for myeloma. As many of you know, multiple myeloma is a highly heterogeneous disease, meaning that every person's disease is different. So each patient should receive treatment specific to his or her type of myeloma. That's the promise of precision medicine, and that is the focus of our research efforts here at the MMRF. With such a high degree of disease diversity, the MMRF is focused on generating and aggregating as much data as possible to capture the full picture of myeloma. The cornerstone of this effort is our COMPASS study, which now has the largest genomics data set of any cancer. We make sure that all this data is available to researchers worldwide to develop hypotheses for new, more precise treatments, and then we push those new treatments into clinical trials. This is our precision medicine model. In 2017, last year, we also announced 3 major multi-million-dollar investments that leverage our precision medicine model. One in immune therapy, one in prevention of myeloma, and one in answering the questions that are most important to the myeloma community. To follow all of our latest research and educational updates, we strongly encourage you not only to visit our website, but also to follow us on Facebook and Twitter if you're active on those channels. Now I'd like to introduce our 3 speakers for today's webinar. Dr. David Avigan joined the attending staff of Hematology Oncology at Beth Israel Deaconess Medical Center in 1996 and became the director of the Hematological Malignancies and Bone Marrow Transplant Program in 1997 and the chief of Hematological Malignancies and Bone Marrow Transplant Section in 2014. He's a professor of medicine at Harvard Medical School. His laboratory efforts have focused on the development of dendritic cell-based vaccines, including a model in which patient-derived tumor cells are fused with dendritic cells as a novel, patient-specific vaccine. Based on these findings, he has supervised a series of clinical trials to examine the immunologic and clinical efficacy of this vaccine strategy for patients with hematological malignancies and solid tumors. Dr. Nina Shah is Associate Professor of Medicine at the University of California in San Francisco, and previously served as Associate Professor at the University of Texas MD Anderson Cancer Center. Her research focus is on multiple myeloma treatments, including cellular therapies and vaccines, and she has been active in myeloma clinical trials. Also today, you will hear from Kirk Kramer. He's a myeloma patient and a participant in a myeloma vaccine clinical trial. Kirk is partner at the Bridgespan Group, a nonprofit that provides advisory support to other nonprofits and non-governmental organizations globally. He serves as the head of Bridgespan's leadership practice, which provides consulting support and leadership development to executive teams of mid-sized nonprofits. Kirk and his wife Kate split their time between homes on the south coast of Massachusetts and the mountains in southern Colorado. They have a son in Boston, another in Philadelphia, and a daughter in Portland, Oregon. Welcome to all of our speakers. Today, Dr. Avigan and Dr. Shah will provide some information on the following topics. They will talk about understanding the rationale for vaccine therapy, vaccine platforms, the rationale for combining vaccines with immunomodulatory drugs, checkpoint inhibitors, and T-cell approaches, and the challenges of translating vaccines and cell therapy into the real-world setting. Now it is my pleasure to turn the webinar over to Dr. Avigan. David, nice to have you with us.
Thank you very much, and it's a real pleasure to be here, and welcome to everyone who's joining. So what I think I'd like to do over the next short period of time is to talk a little bit about how the immune system works in general and how we hope to use this as a powerful force in fighting cancer, and specifically in targeting multiple myeloma. And so in my first slide, really this slide, which is a complicated one, is to just talk about the diversity of the immune system and the different sort of layers of defense that we have in fighting what typically we think of as infectious diseases or other challenges to the body. And the immune system can really be divided into two broad groups, or two broad arms. There is the innate immune system, which is really our first line of defense, and it includes cells like macrophages and neutrophils, whose job it is to kind of provide an initial line of protection by swallowing up foreign things that don't belong, by secreting certain proteins or cytokines that essentially degrade foreign invaders without knowing too much about them. It's sort of like a first line of protection when things create certain danger signals that they react to. And then the second part of the immune system, what we call the adaptive immune system, is a slower but in some ways more long-lasting effect. And this has both teachers and learners. The idea here is that the immune system is sampling things from the outside world, deciding what's dangerous, and when it comes upon something dangerous, it then educates its actors, which are the T cells and B cells, to make proteins, antibodies, or other types of things that will fight against them. And they generate a response that expands and creates memory, so that if you are seeing chickenpox or mumps or other things like that, you develop this first line of defense, the innate immune response, but you develop a more long-lasting adaptive response that consists of B cells and T cells that are essentially raised to fight against this particular pathogen and can provide long-term protection. So that's really the two sides of the immune system, and when we think about trying to sort of use this as a tool against cancer, it's important to remember that. We're going to really focus on the adaptive immune system, which really is the more long-lasting effect of that. So when we look at the next slide, this is sort of a bit of history. The idea that the immune system might be used as a powerful tool against cancer is not completely new. As you see a little bit of a reference from an article from 1900 from the New York Times, and it was based on this sort of observation that frankly people didn't understand, which is that every now and then when somebody who had cancer got a bad infection, their cancer seemed to regress. And it seemed to imply that some kind of runaway inflammatory response of the immune system seemed to also target cancer. I can't say that we knew how to use it well, and as you see in this well understated article, many cases cured here. This ended up not really being an answer in and of itself, but showed a powerful model. And then if you look at the next slide, that is really where in the modern era we began to learn about how the immune system can target cancer. And this is the idea that when we do a bone marrow transplant in certain diseases where you're using a donor's cells, that the donor's immune system that was being transplanted with the transplant was actually the powerful actor that was working against disease and in some cases was protecting patients from relapse. The problem is that the donor immune system is not terribly focused against the cancer. It sort of recognizes lots of things, including things that are not specific to the cancer but are shared by normal tissues of the body. And so you can see in this slide what we call evidence of graft versus host disease, which is an attack against normal skin. And so the idea from a bone marrow transplant was that we had this powerful tool of the immune system working against cancer, but we did not have it really well focused or targeted, or as was referred to earlier, in any kind of precision type way. So let's look at the next slide, which is what we're trying to do now in the new era of personalized immune therapy. And we really have three goals. We want to create an immune response using that adaptive immunity, those learners of the immune system, to target the cancer in a specific way so that it targets proteins or aspects of the cancer that are unique so that it leaves normal tissues alone. You also want it to be broad, and so one of the nice things about the immune system is, as I showed at that first slide, there are many levers to it. There are many different types of cells that cooperate together to fight the disease, and because of that, it makes it harder for cancer cells to escape. And so part of what we want to use when we generate an immune response is some degree of breadth that will target the cancer in different ways, different parts of the immune system, and also not just go after a single protein and target, but potentially go after multiple things. And this is something that I know Nina is going to talk about a little bit later. And then the final piece is the idea of memory, that once you generate a response, we don't want it to be short-lived, but we want that adaptive response where you have long-lasting B cells and T cells that recognize the tumor, that stay in the patient's circulation, and protect from what comes next, meaning protecting from relapse. Now, when we think about the different pillars of immune therapy, there are a couple of different ones, and we're going to focus today on vaccines. One of them is antibody therapies, and in myeloma, examples of this include daratumumab or elotuzumab. They target a specific protein on myeloma cells. They can cause death just by directly binding to the cell, but also they can recruit other immune cells, and there has been some effort to understand how they may interact with other things like vaccines. There are also drugs that we know stimulate the immune system in a more nonspecific way, and an example of that are the IMiDs like lenalidomide, or the new class of drugs called checkpoint inhibitors like nivolumab, but those drugs by themselves, at least from an immune perspective, do not generate a response that is long-lasting, but they may be important kind of partners with other approaches. And then the two main pillars of personalized therapies have been vaccine therapies that we're going to talk about today, which is all about educating the patient's own immune system and specifically their T cells and B cells to target myeloma cells, or the idea of engineered T cells, or CAR T cells as an example, where T cells are taken out of the patient, souped up or genetically modified outside of them, and then reintroduced to act as powerful anti-myeloma therapies. So those are the four pillars. So when one thinks about a vaccine, one has to think about what are the basic dynamics that tell immune response to happen versus not happen. And so we have T cells that circulate in our body, and some of them are against normal tissues like skin, and some of them are against things that come from the outside that are dangerous to us, whether it's cancer or infection. And so we have a group of educating cells, and dendritic cells are the most powerful example of this, that circulate and sort of sit in very strategic areas. They sample things, they decide what's dangerous, and based on what's dangerous and what's not, they will educate T cells to get activated and expand and go after the dangerous things and leave normal things alone. And so that education process, those dendritic cells, become a critical thing that we're going to try and exploit to generate an immune response by now taking those dendritic cells and teaching them to stimulate T cells against cancer. So here you see the different kinds of broad ways that we set up vaccines, and they're sort of designed into either what we call cell-based or protein-based. In a protein-based vaccine, what we're basically doing is we're taking a protein or a group of proteins or antigens, what we call them, from the tumor that we say are somewhat unique to the tumor, and we mix them together with immune-stimulating drugs or what we would call danger signals. They're introduced into the body where they recruit those dendritic cells from the body itself and stimulate a response. The other approach is that we do all of this actually outside the patient, so that we take patients' blood cells, we grow them into dendritic cells so we can make these very active dendritic cells outside the body. We load those dendritic cells with material, and this can be from proteins from the cell, but as you're going to see in our example, we take the patient's own tumor cells, their whole myeloma cells, and we put them together with the patient's dendritic cells. So now that the dendritic cell is there, it has all of those unique parts of the tumor with it, and it can present and teach the immune system to fight and recognize it. Now, one of the questions people always ask is, well, we know about vaccines because we know we get vaccines as a kid, that we are protected against things like measles. What are cancer vaccines? Are they the same thing? And in some ways the answer is yes, and in some ways they are a bit different. The answer is yes in the sense that, like infectious disease vaccines, they are basically engineered to stimulate immune responses inside a patient to protect them. What's a bit different is that infection vaccines are usually done before exposure. We'll take a piece of the flu or a piece of a virus that we're trying to immunize, like the mumps, and stimulate against that so that we're protected from infection. Right now, the way cancer vaccines are designed is that we essentially take the patient who already has cancer, but where their immune system is not active, it's not seeing the cancer as foreign, and we're trying to go into that setting, educate them so they'll make a response to shrink their disease and hopefully prevent recurrence. And when this might happen is something we're still learning about. There is a general notion, I think, that vaccines work best when the disease is first reduced to a low level. So we often partner it with chemotherapy so that, for instance, the disease is first knocked down, and then we use the vaccine as a way to try and prevent regrowth or recurrence. And there's been some special attention to the period after transplant, where during that period of recovery after transplant, there actually may be a greater sensitivity to vaccine because the immune system is just reordering itself, and the vaccines may be particularly potent in that setting. So these are the different ways one might deploy vaccines. So let me stop and turn it over to Nina for the next piece.
Hi, I'm Dr. Nina Shah, and I'm also very grateful to the MMRF for allowing us to participate in this webinar. And we want to talk about some very active areas of investigation and very exciting areas, which include a vaccine platform. So there are some key features of vaccine design, and David went over this a little bit already, but basically you have to have the components of something particular to the tumor and then something particular to the immune system. So there's different ways that you can go about this. Of course, you have to define the optimal target, which in our case would be, for example, a tumor protein or a protein that the tumor expresses, and you have to tell the immune system that it needs to work hard, and that was what we call co-stimulation. So if you look at the diagram on the left, you can see that there are different ways that we can approach this vaccine design. Sometimes people will use a single antigen, meaning a single protein. So that can be in the form of a peptide, which is basically a small protein, or a larger protein, or a neoantigen-based multivalent vaccine, and that really means that it's mutated proteins that are expressed on the cell surface of these tumors. But then again, you can also consider to do something a little bit different as far as your target and look at a whole tumor as a source of antigens. And to do this, you can take tumor cells and break them up and get various sources of materials from a particular tumor cell. One of the advantages of this is that it may allow for a personalized approach, as you'll be hearing from David, because each patient's tumor cell may be different, and we don't know and cannot possibly discover every singular antigen that would be specific to each patient who has myeloma. Once you've decided what kind of tumor target or protein target you're going to work with, you can either consider giving this to the patient as just the protein with some immune stimulant, or you can actually give those proteins to some cells, as David was saying, outside of the body and try to stimulate or load the immune cells and give those immune cells back to the patient so that you can then stimulate the immune system in general. Although it seems very complicated, it really goes on the backbone of what David was talking about, which is that if you have proteins, for example, as we've seen with infections, and you're able to stimulate the immune system, hopefully long-term, the patients can then have a longer-term immunologic reaction to their myeloma. As you can see here, there have been a variety of types of cancer vaccines that have been developed. A lot of these are in early phase clinical trials, and we'll talk about the challenges of doing those and getting a readout of those in the next part of the talk. So we can kind of group these things into cell-based and non-cell-based. When we talk about cell-based, we talk about the fact that these vaccines have to have a protein component, as we were discussing, something to target, and they have to have a component of the immune system, for example, antigen-presenting cells, or as David was talking about, sort of our professional ways of telling the immune system to rev up, or they can be with, for example, a cell that may come from someone else, in the case of an allogeneic stem cell transplant. So when we talk about antigen-presenting cells, or if they're commonly called APCs. These are the immune cells that really know how to show our immune system to activate. And one of the cells that we're very interested in is the dendritic cell, which is the most advanced at doing this. And so you can actually load these dendritic cells with the protein of choice, and then these dendritic cells are capable of stimulating the immune system. You can also fuse these dendritic cells to the myeloma cells, and that's something that David will be talking about in the vaccine trial. These are made from myeloma patients and stimulated outside of the patient, meaning ex vivo, and then injected back. As you can see here, this is an example of the DC tumor fusion vaccine that we're studying in our clinical trial. Here, the patient's own myeloma cells are fused to the dendritic cell. So first we collect the myeloma cells, and then afterwards we collect blood for which we can make dendritic cells. And once we fuse them, literally with sort of a chemical reaction, but we're able to push them together, you can see now this sort of blob-looking cell on the right is a fusion cell. And what it does is it expresses all sorts of different proteins that are particular for this person's myeloma, and it exposes them in the context of being an immune-presenting cell, meaning that it's able to stimulate the immune system now that it's able to express those antigens. So again, this is a way for proteins that we know about and proteins that we don't know about to be expressed on this vaccinated or on this vaccine in order to stimulate the immune system. There are a variety of these cell-based myeloma vaccine trials, and this is not necessarily to confuse you, but just so that all of you can know that this idea has been tried, whether by pulsing or stimulating these cells with a component of your myeloma protein or by using proteins that are commonly known to be expressed on the surface of myeloma cells of all myeloma patients, not necessarily just one particular myeloma patient. And there is not only the ability to stimulate the dendritic cells and give them back to patients, but there's also the ability, as we just talked about, to fuse these dendritic cells and give them back to patients. As you can see, most of these trials have had a handful of patients, and I think that's because everything has been very early phase in studying, and we also want to make sure it's been safe. There's a variety of ways you can give these cells, and some of them can even be given as a subcutaneous injection like you're used to getting with the other vaccines. Regarding the results, it's one of the things that's very interesting about vaccines is that they're not your sort of typical clinical trial medications, right? They're not the kind of thing where you give it and a tumor cell is going to die right away. It's the type of thing that helps the immune system to work better. So although a lot of these percentages are promising, remember, one, that the sort of important thing that we look about is how long patients can stay in remission, and two, a lot of these studies previously had been done in the setting where patients had less access to medications than the ones that we have now, and the more advanced medications that are more available to patients. So this is a moving target, but something that we think is very promising because we really want to make sure that we can have a long-term solution for our myeloma patients. So just to highlight three types of vaccines that have been given some actually very nice clinical results, the MAGE-A3 peptide vaccine sounds complicated, but it's a pretty simple idea. They're using proteins that are presented by patients on their regular myeloma cells and using them to generate T-cell responses. And what they could see, without going into too much of the detail, is they actually had a pretty nice long-term survival, and also the T-cells were shown to be expanded. So if you're able to look at a patient who's gotten the vaccine or not gotten the vaccine, you can see that patients who get the vaccine tend to have their T cells more revved up to fight against myeloma. Similar results were seen in a very small study of something else, another protein called WT1, same thing, where patients got this in the setting sort of a transplant, which, as David mentioned, is a very important time where the immune system kind of resets itself. And so a lot of these trials are given in that setting. And here they were also showing that patients who had very high-risk disease and got this vaccine in the setting of a transplant were able to have nice long-term outcomes. And because of that, they're going to do a phase 2 study, probably in the transplant setting. Finally, something interesting that has been looked at is this PVX-410 vaccine. And this looks at a couple of proteins that are known to be expressed on myeloma patients, particularly in the setting of certain kind of T cells. And these studies have been looking at smoldering myeloma patients who are at high risk of progressing to active myeloma. And I want to point this out because maybe, as it turns out, smoldering myeloma may be one of those ideal times to catch the patient because they haven't quite developed clinically significant myeloma yet, but have a risk to. And therefore, we're trying to see if this can be prevented or delayed. And in this smaller study, they did see an immune response to this vaccine, and this was enhanced by Revlimid. And as many of you know, Revlimid can enhance the immune system function. So this seems like a nice combination for these patients. And as I'll mention, there's a future trial planned. So what about ongoing clinical trials of myeloma vaccines? This is extremely important because there are so many and it's hard to differentiate which one is better or worse or more appropriate. The real answer is that we don't know yet. Each of these particular approaches has the possibility of increasing the immune system activity against myeloma, sometimes in combination with a drug that you would be taking anyway, like Revlimid, and sometimes in the absence of that. Many of these trials will be looking at giving them during the stem cell transplant recovery, which, as we mentioned, is a good time to sort of reboot your immune system and retrain it so that it's able to then fight against myeloma and, more importantly, remember against multiple myeloma, as you can see. And just as I mentioned about that smoldering myeloma trial, there's one that's actually in combination with a medication that's called cytaranistat, or as it's known to other people, some HDAC inhibitor. And so it's looking to see if combining this vaccine with that drug can actually enhance the ability to prevent development into clinically significant myeloma. David, do you want to take it over?
Sure thing. Thanks, Nina. So I just wanted to say a word about combination strategies and why I think for many of us in the field of immune therapy, as the field has sort of matured and we've learned more, we really feel like this kind of approach is probably where the future lies. Just to talk a little bit about the sort of basis of the trial that Nina and I and a whole group of folks around the country are working on. This has to do with this dendritic cell fusion vaccine, and we talked about this already as a bit of a personalized approach where we take the patient's own tumor and we fuse it together with their own dendritic cells. So it's a very patient-specific, personalized approach. And we think that one of the strengths of this approach is that because we're using the patient's whole tumor, that there are going to be responses against a variety of antigens, a variety of targets that are going to be broad. And we'll also capture different subpopulations within the tumor, because we know that part of the challenge is to not just focus on a part of the tumor, but to try and avoid escape by hitting the whole tumor in unison. So this is data from an initial study that we did at Harvard where we essentially vaccinated patients after transplant. And there are two things that we saw. On the left side with the green bars, you can see that the number of immune cells or T cells that recognize the patient's myeloma considerably expanded and continue to expand during the period of vaccination after transplant. And this seemed to correlate with a near doubling of the number of patients who achieved complete remission after transplant, really without any other therapy. And that really laid the groundwork for what became a national approach that I'll talk about. Now, I think the other piece, as we said, is that one of the concerns about vaccines is that we're stimulating and trying to educate the patient's own immune system, but we know that the immune system in cancer has potential defects. In fact, that may be part of the reason why the tumor is allowed to grow and expand. And so at the same time that we're trying to educate this immune response, we have to understand what are those other pieces around the tumor that create an immune suppressive effect, and how might those get in the way, frankly, of our vaccine. And so you'll see that a lot of the combination strategies involve some kind of education approach or CAR T-cell approach, along with another approach that helps to repair some of the basic aspects of the immune system, whether it's things that are suppressing T cells or cells that are suppressing things, and so that's going to really be a focus. So this is a schema of our large national trial that we're doing right now with the fusion vaccine, and it basically involves taking patients after transplant. So we said that that's a unique time where the immune system is just rebooting itself and is really more susceptible actually to vaccines. And we combine that with lenalidomide, which we know is a drug that we often give after transplant, and we think part of it has a direct effect against the myeloma cells, but it seems to also potently stimulate the immune system. And so here what we're doing is we're taking this unique setting after transplant, we're adding the lenalidomide as a way to try and boost immune responsiveness, and then in that context we add the vaccine. And so in this trial, patients are going to be randomized to either undergo the vaccine plus lenalidomide or get the standard, which is lenalidomide alone. And this is a study that I have to say was a pretty bold adventure because it involved teaching sites around the country how to make our vaccine, and they all came to Boston, and we ran a cooking show and taught them how to do this in an open source format. And we have 17 sites that were ultimately enlisted, and we really created a communal effort to generate this personalized vaccine. And we've now enrolled almost 200 patients in a little over a year, so it's been really a remarkable collaboration, and I think has shown very importantly that we can do personalized therapy on a larger scale when we all work together. There have been other approaches. The GVAX approach is also cell-based approach where myeloma cells that come from a patient are altered so that they then stimulate responses because we put in a danger signal into the myeloma cells, and that's also now being studied in combination with lenalidomide as another whole cell vaccine with the approach to try and generate a more durable response. Another exciting area is combining vaccines with checkpoint inhibitors. Now, checkpoint inhibitors have become quite prominent in cancer therapy. They basically undo one of the main ways that cancer cells hide from the immune system, this so-called PD-1, PD-L1 pathway. And we know that if you block that pathway, tumor cells become more recognizable. But when that was used by itself in myeloma, it was a bit of a dud, and the reason for that is that there probably aren't a lot of T cells there to activate. So our idea and others has been the idea of using a vaccine, in our case the fusion vaccine, to stimulate the T cells and then to use the checkpoint inhibitor to further amplify their response and activate them. Again, this sort of dual approach that hits different parts of the immune system at the same time. And then the last thing I wanted to end on is the idea of combining CAR T cells and vaccines. CAR T cells have gotten a lot of attention because of some really exciting data that show that they have effectiveness in certain diseases like leukemia, lymphoma, and now being looked at in myeloma. And what they are cells that are extracted from a patient, changed genetically so that they now recognize a piece of tumor, and also activated by putting in certain co-stimulatory mechanisms into the T cells. So these T cells are souped up, and they are fantastic killers. But one of our challenges with CAR T cells is making sure that they hang around, because these cells can be very potent, but then they can disappear. And we see that there is a pattern, at least in some patients, even though they have nice responses, that they then relapse. And the idea was vaccines have a weakness in that they need T cells in the body that can respond to them and be active, like CARs. And CARs have a weakness that they need to be stimulated so that they don't disappear. And so through a really fantastic effort that the MMRF is spearheading and funding, we are working with groups at Sloan Kettering and in the Netherlands to put together a combined strategy of looking at CAR T cells and vaccines together and to see whether this can lead to a long-lasting immune protection in the setting of myeloma. So I'm going to stop and hand it back to Nina.
Great. Thank you, David. So with all of this excitement, we have to also remember that there are challenges in the real-world setting, and we're so grateful to many patients like you guys and the MMRF for supporting efforts, because without patient participation, it's really hard to get forward in this very exciting field of study. Just want to talk about some challenges in application in the last minute or two. Remember that there are some limitations. Now, normally when you get a vaccine, it's a young, healthy baby, or maybe you're getting a flu shot and you're healthy, but in the setting of an immune-compromised patient, for example, a myeloma patient who by definition will have immune compromise either from their disease or maybe because of their treatment, you need to make sure you sort of reverse that host immune dysfunction. And for that, we talk about things like adjuvant, and things like that would be considered, for example, the GM-CSF you've already heard about. And then this issue of timing. As David already mentioned, we're not sure whether we should be giving this in a smoldering setting when sometimes people aren't even diagnosed yet, or maybe after they had some remission, or perhaps, as we've been trying to do, after transplant when the immune system is regenerating. And when you have a lot of tumor burden, you may think that's not the time to give the vaccine. And then other times, if you think you don't have enough immune cells because you've had so much tumor eradication, you don't know if that's the right time either. So that's always a moving target. And then I think one of the biggest challenges is finding the right combination of antigens to target. And it's not clear whether this is going to be the general myeloma proteins that we always have, or if it's going to be special myeloma proteins that each patient has. And so this is something also that we're following. Now, I think one of the biggest challenges and the reason why we need participation from all of you is showing a difference. Remember, the myeloma vaccines tend not to be a treatment, but rather a way to boost the immune system to make a difference for longer-term survival. Myeloma is a marathon for a disease, and we want to make that marathon as long as possible. And it's hard to show, for example, efficacy results without having longer-term follow-up. So we really push for each other who are trying to get these trials done to really show a difference in progression-free and overall survival, because ultimately that's the metric that we really care about, and that how the immune system will work. And of course, that's more difficult to do with all the great drugs we have, but I think it's definitely worth pursuing. And with that, we'll move on to our patient experience.
Great. Hi, my name is Kirk Kramer, and my patient experience is based on participation in the primary vaccine trial that Dr. Avigan just discussed. It's this phase 2 trial that fuses dendritic cells and myeloma cells, and having that goal of educating, in this case, my immune system to attack and find my myeloma cancer cells. A little bit of background. I was diagnosed with multiple myeloma in October of 2016 at age 61. Came to the hospital with what turned out to be a pathological fracture caused by thinning of my bones due to multiple myeloma. I was lucky enough to get referred to Dr. Rosenblatt at Beth Israel Deaconess Medical Center, who's a colleague of Dr. Avigan's, and they've got a great team there and obviously a lot of experience with these vaccine trials. When I was diagnosed, I was offered the chance to participate in this trial and needed to make a fairly quick decision. That was because a bone marrow biopsy was required to extract my myeloma cells before I could really start treatment for multiple myeloma. A couple of factors that kind of convinced me to participate. And first of all, it was a phase 2 trial, and there were really positive results from, I think there were around 70 patients in the first phase of the trial. So I already had some information about its efficacy. Second, the science behind this is just incredibly compelling. This idea of creating a vaccine really for my own cells, both my cancer and my immune system cells. So you couldn't have a more personalized treatment. And third, my wife has a PhD in genetics and was teaching human genetics at Brandeis University. And not only has she been a great supporter throughout my treatment, but along with Dr. Rosenblatt, really helped me understand kind of the potential benefits and upside of a vaccine. And I guess the final thing here is this is a random control trial, so there was 50-50 chance of getting the vaccine, but in any case, even if I didn't get the vaccine, I was going to be getting the same great treatment that I otherwise would have received. So there was really no downside, just kind of all potential upside here as I saw it. In terms of treatment process, it was really pretty standard until the point that I was selected for the vaccine. I think the only thing that was really perhaps additional was I did have that extra bone marrow biopsy up front to obtain the cancer cells, which were then frozen in case I got in the vaccine trial and would have the vaccine made. But I went through standard cycles of Revlimid, Velcade, dexamethasone, 21 days on, 7 off. and was lucky enough to have some pretty quick positive results from that. I then had an autologous stem cell transplant in July of 2017 after a few delays. And following that transplant, and even through today, I've been in either stringent complete response or complete response, so good results there. Around day 60 after the transplant is when I got the news that I was randomized into the vaccine group of the trial. To create this vaccine, I had one extra procedure, another apheresis procedure to remove blood cells, white blood cells, to be able to get the dendritic cells. And then the vaccine was fusing these dendritic cells and the multiple myeloma cells that had been taken in that biopsy back in October 2016 and frozen. Around 100 days after the transplant, I received the first of 3 injections of the vaccine. Appropriately, the first one just after Thanksgiving, and then the next 2, each a month apart, so presents for Christmas, and my birthday happens to be in late January, so I saw them as great presents all around. I guess the surprising thing to me is the injections were really quite small in size, but amazingly, each contained about 2 million cells or so. Following the injections, I had to give myself shots for around 3 days around the site to stimulate white blood cells to the site of the vaccine injection. And frankly, the only side effect I had was really pretty minor, kind of rash and itching around the injection site for about 7 to 10 days or so. As Dr. Avigan mentioned earlier, the trial is a 2-year trial, and part of the trial is taking Revlimid along with the vaccine. So I'm taking 10 milligrams of Revlimid daily. You know, I see Dr. Rosenblatt and team monthly and have tracking tests done. And my sense is the trial doesn't add a lot to my treatment. I may have some extra blood drawn for tests from time to time, but really nothing major in addition to what I normally would have been going through. So overall, this has really not added a lot to the normal treatment process, and the side effects of the vaccine have really been quite minimal. The last thing I'd say is, should you participate in a immunotherapy vaccine trial if offered the chance? Of course, I can really only talk about my own experience, which has been quite positive. But as Dr. Avigan and Dr. Shah discussed, I think vaccine therapy is really a very promising concept, providing personalized treatment by using an individual's own immune cells to attack their cancer cells. Reflecting on my own experience, I think if you're offered this kind of opportunity, maybe a few thoughts on making a decision to participate. Obviously, what phase the trial is in can matter. If you're in phase 2 or later, you can fortunately learn from earlier phases, both the results and the risks. I think also quite important is confidence in knowing if your doctors and hospital have experience with this or similar trials, and I suspect this would be particularly important if you're considering a phase 1 trial. In this case, I knew my doctor, Dr. Rosenblatt, and the team, and obviously Dr. Avigan at Beth Israel had been involved in many vaccine trials and are highly experienced with this. One thing about random control trials of this type is you do have to be willing to bet on the upside. The reality is you have to recognize that you won't know what your individual results are from the treatment, since the results will really be reported only at the trial level overall. So while I believe it is promising for individuals, really I will not know how it works for me. But I do think that participating and contributing to help advance research is really important for all of us. It helps others, and hopefully in turn will help us as individuals. And I'm quite grateful to individuals in phase 1 of this trial who gave me the opportunity to participate in this as well. So thanks for letting me share my experience with you, and I'll turn it back to Mary for the Q&A portion of the webinar.
Great. Thank you so much, Kirk. That was fantastic. And also, thank you so much, David and Nina, for your presentations. And now we're going to move into the question and answer portion of our program. So as a reminder to everyone, if you'd like to ask one of the presenters a question, just type it into the Q&A box and click Enter. and your questions will be answered in the order in which they were received, but due to time constraints, we may not be able to answer all of your questions. So I have to say, Nina, that your call to action here in the presentation for more people to join vaccine trials has clearly been heard, because many of the questions that we received were around how patients will know that they're eligible for these trials and which patients are, good candidates for vaccine trials. And of course, this is something that patients would need to discuss with their doctors. But I mean, in general, would you be able to enter a vaccine trial if you weren't eligible for a transplant in the first place?
Yeah, that's a really great question. So although I mentioned that many of these happen in the setting of transplant, there are a few that do not happen with transplant. For example, the smoldering myeloma doesn't, although if you had active myeloma, you might be looking at something different. Each trial is so different that it is worth doing a little bit of research, which you can do on clinicaltrials.gov, and asking your provider if he or she knows about that particular trial. It's not necessary that all of the trials are going to have transplant, and if you don't think that you're a candidate or your doctor has said you're not a candidate for the transplant, it's still worth pursuing those trials that don't have transplant as a component.
So would you be able to be a candidate for a vaccine trial if you were a patient who was like a relapsed refractory multiple myeloma patient who had already had a transplant some time ago?
Can I jump in for a sec on that one?
Go ahead, David.
Yeah, definitely.
Yeah, no, I mean, I think as Nina said, each one of these studies is unique, and I think that the setting that they're exploring is going to be different, and what might be available in a particular center, in a particular geographic region of the country may have some, differences as well. We are embarking at our center at a trial involving our vaccine with a checkpoint inhibitor in the context of relapse disease. So that happens to be just an example of a study that is in the setting of relapsed, not transplant, that is asking a question like that. I think we do have an overall sense that each of these immune therapies have different strengths and weaknesses, and that vaccine therapy is probably going to be best mixed with some kind of cytoreduction, meaning reduction of the tumor prior to doing the vaccine. And so I think ours and various other strategies that are asking the question when there's more disease around are combining it with other approaches as well.
So, Kirk, here's a question for you. And one of our listeners heard that you were talking about taking Revlimid in conjunction with your vaccine treatment, and they're asking whether you have to take dexamethasone as well.
In this case, I don't. So simple answer. Maybe Dr. Avigan knows why I don't have to.
Yeah, I mean, so first of all, it's a great question, this notion of, we know that dexamethasone or steroids in a broader category are very effective in myeloma therapy. They're often partners with some of the treatments that we do. And yet, when you're stimulating the immune system, there is at least the potential that steroids would get in the way of that. In this case, it was sort of easy because when we used lenalidomide maintenance after transplant, we generally don't give it with steroids. There might be some examples where people do, but I would say often we don't. So we were using the backbone of maintenance therapy, which is with standard lenalidomide but no steroids, with our vaccine. But it has come up, and I think that the answer to this question is not totally known. There is at least the idea that steroids may be a little bit counterproductive if you're combining them with a vaccine or with, for instance, CAR T cells.
So here's an interesting question. One of our listeners asked about plasma cell leukemia, which is a similar disease to multiple myeloma, and wondering whether these types of vaccine approaches are being used for that disease. David or Nina?
Yeah, I can answer. I think one of the most difficult things about plasma cell leukemia is, it's known to be an aggressive disease, and a lot of times clinical trials will exclude those patients. So it is a trial-by-trial basis. However, we are really trying to push in the myeloma community to expand these inclusion criteria, and I'm really glad for patients like you to point this out to us. So hopefully that's going to be more and more available for patients with plasma cell leukemia. There's really no reason in my mind that a patient with plasma cell leukemia should be excluded from a vaccine trial if they meet the other eligibility criteria. So we're hoping to make that more available in the future. David, you want to say something about that?
Sure. No, I think as you said, there are many similarities between plasma cell leukemia and myeloma. They probably are on somewhat of a continuum, and so it would be wonderful to have them incorporated, those types of patients incorporated into studies so that we can learn more about what works well and what doesn't work well. I think that, again, there is a notion with immune therapy sometimes that if the disease is growing more rapidly, that treatments that take a little bit of a longer time to have an effect may not be as helpful, but we don't know that. And I would say in a broader way, some of the risk components that we think about, we know that certain genetic changes are associated with a higher risk profile when we think about drugs. Does that apply to immune therapies such as vaccines or T cells, or is it different? We don't know, and we won't know unless we study it. So I think it's important to be inclusive.
And here's a question, David, about the level of risk for patients that may be candidates for vaccine trials. So when you're looking for patients to enroll in your study, are these studies, these vaccine trials, really meant more for patients who are sort of defined to be more of a high-risk population, or are they really more for patients who cytogenetically may be more of a low-risk population?
So I think we honestly just don't know the answer to that question. And we can come up with sort of ideas and theories, but the only way we'll really know is through the studies themselves. So again, I think that these different types of immunotherapy have different sort of features to them. So vaccines, which take a little while, just in the same way you think of getting immunized, for instance, against hepatitis or tetanus, you often need a booster shot. It takes a little while to develop your response. There may be some advantage to looking at that in patients where the disease is at a lower, sort of level, or when it's been reduced first, as opposed to, for instance, CAR T cells that may be more effective when they see disease around and react to it, so to speak. But in terms of whether the categories of risk apply, you could really theorize in different directions. For instance, patients with more genetic abnormalities that we know have had, higher risk, let's say, profile with regular drugs, those may also create better targets for the immune system. So I think the idea is that we have to study these different groups, try and learn as much as we can, and see what we can do to be helpful to the different, sort of subsets of patients. But honestly, until we do the studies, we don't know the answer.
Right. I mean, it's still rather early days in this in the vaccine arena for myeloma. So there's been a couple of questions about MGUS. So, I mean, you were talking about vaccine trial or trials that are being done in the smoldering myeloma population, but there are a couple of people who are asking whether or not there's any type of that vaccine trial activity for patients who are MGUS.
To my knowledge, there isn't an MGUS-specific trial yet, and the reason for that is that we don't know. So, for example, many of these people never progress, and even those that are high risk to progress, it may take a couple of years, and so it's really hard to measure the efficacy of this. Now, that doesn't mean that it's not worth measuring, for example, T-cell expansion or other things like that, but my knowledge so far, I think it's only smoldering. David, do you know of any MGUS?
Yeah, no, I think my understanding is exactly as you said. I think one of the challenges when we think about therapies for patients at earlier stages of disease where the standard of care might be to leave them be, is, there's always this sort of, I would say, temptation to want to jump in and intervene. And of course, we want to do that if it makes a difference. we also want to be careful that we're not doing stuff or making patients jump through hoops when it doesn't actually add that much. And so I think that they have to be, that kind of stuff has to be thought about in a carefully designed way. And I think, as Nina said, the challenge is if you have a large group of patients who are just going to be fine anyway, you want to make sure that either you're defining really the high-risk group there, or you have a way of measuring your effect so that you've learned something and know whether the next group of patients are going to benefit or not.
So here's an interesting question, and this person, maybe they're a double dipper, but they asked if they've already had a vaccine, a myeloma vaccine, would it be possible to enter another trial and have a different myeloma vaccine?
Actually, David, do you want to answer that?
No, go ahead.
So some of again, this goes on a trial-by-trial basis, but some of the trials exclude previous quote-unquote cellular therapy or other vaccine therapy, but some of them will say they exclude it only within a certain amount of time. So this is not a blanket statement. You really have to look at the individual trial that you're considering with all the inclusion and exclusion criteria.
And is there any... Oh, sorry. Go ahead, David.
All right. I just wanted to mention one thing that I think Nina spoke about earlier as well, which is this sort of notion of how do we measure the effect of this, and how might treatments that are given sequentially even affect each other. I think that many of us in the immunotherapy field have observed, at least in individual patients, a disease that has not disappeared or even has come back in a way where it requires treatment. But when you look down the road over a 5 to 10-year period, the course seems to be different than what we would have expected and much slower. And again, one has to be careful that one isn't just sort of like reading into things that aren't there or wanting to see something. But I think that these treatments given sequentially may affect each other. There's been that experience with, for instance, when checkpoint inhibitors were given to certain patients with solid tumors who got a vaccine and didn't seem to respond, or didn't respond for a long time, all of a sudden they went back into response because the immune system was kind of reactivated. So I think there may be ultimately a knowledge that we have that these things interact in a way where sequential therapies actually build on each other. But the bottom line is, we're still in that learning phase. And the practical question of does it exclude you or not, as Nina said, depends on the specifics of each trial.
So, and is there any blanket exclusion criteria for patients who may have been or are currently on Amplicity or Darzalex, one of the monoclonals that are currently approved?
No, there's not a blanket, because now we know that many of these people, have had this therapy. Sometimes what they'll have is a washout, so that you can take the drug, but you have to have a couple of weeks before you're able to be part of the vaccine trial or get the vaccine. So it's not a blanket thing. I mean, of course, every trial is different, and you have to check, but I don't think there'd be a reason to exclude people for that.
Yeah, and there may even be ways that they build on each other. There's some effect that some of the antibodies may have in stimulating the immune system in a more general way. And so I think, as Nina says, I think it's protocol specific, but in general there's no blanket issue that people couldn't have had that before.
Okay, I think we have time for just one last quick question, and this is a person who wrote in about Waldenstrom's macroglobulinemia. Are there any vaccine trials on the horizon for those patients?
I don't know of any, although I am not a self-professed Waldenstrom's expert.
Yeah, I'm not aware of one, although, as Nina said, they often, there's a little bit of a different clinical realm for them in terms of where the trials are, so it's something to look up, but I don't know.
Okay, so thank you so much. That's all the time we have today for questions, and I appreciate your answers. On behalf of the MMRF, I'd like to thank David, Nina, and Kirk for their presentations today. I'd also like to thank our sponsors, AbbVie, Amgen, Bristol-Myers Squibb, Celgene, Genentech, Janssen, Carrier Farm, Novartis, and Takeda Oncology. And also, thank you so much for all of you who have joined us today on the webinar. We do have a number of upcoming webinars for the fall. We are starting off in the beginning of September with a webinar around evaluating treatment options for patients with relapsed refractory myeloma. Then we're going to have a webinar in October that's going to be in support of caregivers. And then in November, we're having a webinar on African Americans and myeloma and their disease risk. So for more information on that, please go to our website. We have a number of live patient education programs coming up in the fall as well. In the middle of September, we're going to be in Nashville, Tennessee. And then in October, we're going to be in Washington, D.C. And then in November, we're going to be in St. Louis. So if you have some interest in these, please go to our website and register, and we'd be very happy to meet you there. And in conclusion, we have many resources here at the MMRF to help you keep your disease journey on the right path. We do have a very knowledgeable nursing staff at our patient support center, and you can call them to ask them any types of questions about your myeloma, and they'll be happy to answer them. And again, we encourage you to stay connected with the MMRF. Please visit us on our website, visit us on Twitter, and visit us on Facebook for continuing updates on the research that we're doing and for education in myeloma.